My name is Ben Kilbride, and this is my presentation about protein kinase associated receptors, how they function and their structure. Protein receptors may also function as protein kinases. Kinases are just enzymes which regulate the activity of proteins. This activity is initiated when they are bound to a specific lignin, and their signals are transmitted through phosphorylation events in the cell. This is a figure of a protein receptor. The big yellow piece is the receptor, and you can see the signal is coming in from outside of the cell, and then the cytoplasm is inside the cell where the signal is transmitted. There are two main categories of receptor kinase. One is one which phosphorylates a tyrosine residue, and another is one which phosphorylates a serine or threonine residue. Receptor kinases are very important for a cell. Some of the things they're responsible for is cell division, cell specialization, cell movement. They're initiated by growth factors, which could be insulin, epidermal growth factors, and nerve growth factors. An important kinase I will be talking about is called receptor tyrosine kinase, and it is a receptor for a little bit more about growth factors. They bind with receptor kinases, which you can see in the photo. They are the red Y-shaped object, and growth factors are like the triangle ones that are labeled signaling molecules, and they are found in blood serum, and they're basically just messengers which stimulate various growths. A specific type of growth factor is called platelet-derived growth factor, or PDGF, and it is secreted by platelets in blood. And you can see the platelet is the whitish yellow object in the center there, and they stimulate the formation of new connective tissue. And the platelet-derived growth factor is recepted by the receptor tyrosine kinase. In order for the RAS pathway to be activated, it uses SOS, which is a GEF or guanine nucleotide exchange factor, which you can see in orange in the figure. And it allows a GTP molecule to join, seen in the active RAS, it's the red molecule, and a GDP is released, which is the other red molecule. And at this point, RAS has been activated. Just as important is the inactivation of the RAS pathway, since it cannot be continuously stimulated. Um, Basically is the reverse, and GTPase activating protein helps with the hydrolysis of GTP, and it releases the GTP, and the GTP comes back to replace it, thus inactivating it. Now, some receptors can have a mutation, like the one on the very right, which is labeled. It doesn't have a tail, and therefore it cannot transmit a signal. Um, while normal receptors undergo autophosphorylation, mutants do not, even though they still allow binding of ligands. And this results in uh, non-functional receptors that do not transmit signals to the cell. The first question is, each of the following is a characteristic of camodulin except A, it can activate specific kinases and phosphates. B, all are characters of camodulin. C, it binds two calcium ions at each end of the protein. D, it has a very high affinity for calcium ions and binds calcium ions irreversibly. And E, when activated by calcium ion bonding, it wraps around proteins that have camodulin bonding sites. The correct answer is D. It has a very high affinity for calcium ions and binds calcium ions irreversibly. In the figure, you could see that it clearly activates kinase, and there are two green dots on each end, which are the calcium ions, and it is also wrapping around proteins. So the only incorrect one is D. Question 2. Each of the following is a G protein coupled receptor or type of GPCR except A. Opioid receptors, B. Thyroid stimulating hormone receptors, C. Beta adrenergic receptors, D. Steroid hormone receptors, or E. Olfactory receptors. The correct answer to this question is D. Steroid hormone receptors. In the figure, you can see a steroid receptor, and it is not coupled with a G protein. So this must be the right answer because it is not G protein coupled. The next question is, each of the following is a second messenger except A. Cyclic adenosine monophosphate, B. Epinephrine, C. Calcium ions, D. Inositol triphosphate, or E. Diacylglycerol. And the answer to this question is B. Epinephrine. In the figure, you can see that epinephrine is coming from outside the cell, and second messengers are a result of a pathway, so they cannot come from outside of the cell, and therefore it cannot be a second messenger since it's coming from the exterior. Next is asking about phosphatase enzymes. Do they A. remove phosphate groups, B. add phosphate groups, C. always inhibit target molecule activity, B. bind tightly by wrapping around a target molecule to regulate its activity, or E. metabolize glucose? And the answer to this question is A. remove phosphate groups. In this chemical equation, you can see that with the addition of phosphatase, the phosphate group has been removed from the substrate. It has not added it, and yeah, it has removed it and replaced it with an OH. The final question is, protein kinase C, or PKC, activity is involved in each of the following except A. increase in cellular pH, B. ion channel regulation, C. PKC is involved in all of the above, D. secretion of proteins and other substances, or E. cytoskeleton changes. The answer to this question is C. PKC is involved in all of the above. You can see the PKC in the figure and how it is regulating ion channels. You can see the calcium ions are relating to the channels, and this increases the cellular pH. So the answer is all of the above for those two.